कितने दिन की? 1 मंथ। मेडिसिन में कोई केस देखने को मिला मेनिंजाइटिस का? कुछ नहीं मिला। आप सबकी मेडिसिन में पोस्टिंग होगी या पीडियाट्रिक्स में भी किसी की हुई? नहीं, कांट हियर यू, से लाउडली सो आई कैन हियर यू। हाँ, उनको बोलो क्लास ले रहे, क्लास के बाद बात करें जो भी हो। पीडियाट्रिक्स में कोई बच्चा कभी फीवर के साथ देखा? देखा। क्या देखा उस पे बच्चे में, क्या था फीवर का कॉज? पीडियाट्रिक्स में बच्चा देखा फीवर के साथ में, कैसे एवेलुएट किया उसको? मतलब if this is the situation, you cannot and nobody in the class can answer how a child with fever was evaluated, then it is really worrisome. Who has, now raise your hand, who has done the pediatric posting? Raise the hand, otherwise go out of the class. You have never seen a child with the fever in the pediatrics? You have seen, so please get up. Tell me now, child had come with the fever, what else you had looked for? So what were the other symptoms? Okay. Okay. So suppose this class is for meningitis. What you have seen for meningitis, what is in your mind that if a child presents with meningitis, what will be the symptoms? Headache and neck stiffness. Okay. What age group of child you are talking who will tell you that I am having a headache? More than 10 years. So, in your 1 month posting in the pediatrics? 1 month. All the children were above 10 years of age? No. So, if a younger child comes to the OPD, suppose it is a meningitis case, what will be the symptoms? Difficulty eating and speaking, you are saying? Fever will be there? Fever will be there. So, if we say these are the, as per current discussion of ours, these will be 3 symptoms: fever, speaking difficulty and eating difficulty, these 3, nothing else. There will be many. Okay, so we will come back to you. Who has done medicine posting? Just please get up. No, if you do not get up, everyone can leave the class. Who has done medicine posting? Raise your hand. You have done medicine posting. Who else has done posting with you? Entire batch has come. So, I want everyone to raise the hands. Otherwise, everyone can leave the class very well. Now, you sit down. Madam, why you were hesitant to raise your hand that you have done medicine posting? No, it has to be answered. The classes are two-way communication, and I am not interested in having people sitting silently here. Can't hear you. No, I am not asking you, sorry sir. I am asking you why you have not raised hand. If the classes are meant, you have lots of YouTube videos, everything is there. Why we are wasting our time here? Why don't you leave the class then? Please leave. Leave the class. No, I am not interested in your sorry or anything. Please leave the class. I don't want people sitting here just wasting their, it seems you are wasting your time. No, no, please. Next, you, mister. A person suspected case of meningitis will present with what? Medicine means we have already excluded pediatrics age group. So, headache is one. What else? Might have seizures. What else? Okay. Sit down. There could be, no, I can't hear you. Inflammation of, is it called inflammation? Why you will have swelling in the neck? I am yet to come across where the CSF has accumulated to an extent that it is causing a swelling in the neck. No. So, what I am seeing here, that even though you have spent 1 month in the pediatrics, 1 month in the medicine in a hospital which is bursting to its seams, still there is a problem in knowing the symptoms of a patient who may come with a suspected case of meningitis either in the medicine or in the pediatrics. So, it is time that we should not just waste time by standing in the wards rather than to understand what is a case of meningitis presents. So, the topic of this class is primary pathogens. If we say primary pathogens, means though we will focus on the most common causes of bacterial pathogens, because this is bacterial meningitis, we should certainly know what is the spectrum of pathogens which cause meningitis, and then we focus into bacterial meningitis and 3 most common pathogens. So, now next, what are the pathogens which are responsible for causing meningitis? Let's say loudly. Haemophilus influenzae. Okay. Better way of telling is, if you classify bacterial causes, viral causes, fungal causes, parasitic causes, so on. Otherwise, it is not systemic way of learning. So, you are talking about bacterial pathogens. Okay. So, first bacterial pathogen you said is Haemophilus influenzae. Next, I will not put a Klebsiella pneumoniae so high in the list. See, one thing is that every pathogen can be attributed that this will cause meningitis, but that is not the way. Listeria, okay. If I ask you 3 most common pathogens, then you go to Listeria, E. coli, everything can come. No. Now, if we continue this discussion, so the list is not as per the priority list. Okay. So, first you have to have which are the pathogens, primary pathogens. So, primary pathogens for bacterial meningitis is Neisseria meningitidis, even then commoner than that is pneumococcal and Haemophilus influenzae. These are the 3 most common causes of bacterial meningitis, and then your list of E. coli, Listeria meningitis, they are all there. Anything and everything can cause under certain circumstances, but your bacterial list primary pathogens should be these 3. Okay. So, now we are done with bacterial list more or less. Which other class of pathogens? Viral meningitis. Is it common or no? Viral meningitis is common or no? Okay. What other class of pathogens? Fungi, okay. Parasites. So, every class of pathogens you mean to say can cause meningitis? Okay, fine. The answer is not wrong, but if I now want you to list which class of the pathogens are most common? Bacterial. Okay, I will now see the slide. Viral meningitis is the most common cause of meningitis. Bacterial meningitis is most dangerous type of meningitis because it can be fatal within 24 hours. Fungal meningitis is little uncommon and difficult to diagnose, and the most common pathogen in case of fungal meningitis will be Cryptococcus. You have parasitic and amoebic also, though the amoebic will come, and that is why it is subheading. Amoeba cantonensis. Recently, you might have seen a lot of, there was something, Gnatostoma spingerum was isolated in our hospital from an eye, this worm. So, that is also can cause meningitis. And you have amoebic meningitis, especially Acanthamoeba and Naegleria fowleri. So, these are common cause of meningitis, and there is no class of pathogen which we can say does not cause meningitis, but then we should remember in the order of commonality and their seriousness, right. So, these are classes of pathogens. Now, since the class is on primary bacterial pathogens, we will focus on meningococcal bacteria, pneumococcal bacteria and Haemophilus influenzae. You have heard these names or no? Yes. So, coming down to the symptomatology. So, a patient may present with fever, headache, vomiting, stiff neck, aversion to light, photophobia, drowsiness, joint pain, or a person may come with the complaints of having fits. So, these all of them can be symptom in isolation or with conjunction with one or another symptom. Now, the children, why it is important? Because the symptoms can be very deceptive in a pediatric age group, like fever is there, but you may have hand and feet may be cold. A child may be just blank, staring. It could be just listless, refuse to feed. Just child is drowsy. These are the symptoms which are little different from the presentation by an adult population, and that is why if child is giving you a blank look, and if you say no, you have a fixed notion that unless until child is vomiting, having a headache, it is not a case of meningitis, then you may be very much mistaken. So, a child who may present with any of the symptoms, somebody may have a blotchy skin, petechiae may be there, doesn't like to be touched, what we call is fretful, cry is high pitched. These are all symptoms because, and it should arouse in your mind a doubt, am I dealing a child with the meningitis, because these diseases can progress very fast and can lead to mortality. So, that is why you have to act very fast. So, these are the presenting symptoms in babies and toddlers in the new pediatric population. Now comes to the laboratory diagnosis. We have more or less a broad idea I have given you what are the pathogens. So, now I wish to know from your side, in a case of meningitis, what will be choice of samples. So, very nicely answer is coming, CSF sample. That is your first choice of sample, fine. What other samples? CSF is one, no doubt. Blood samples, means blood culture you want to suggest, okay. What else? Nasopharyngeal swabs, always, or in some certain set of population? Certain set of population. Which set of population? No, I can't make out. Say again, please. But in most of the cases, meningitis, the primary route is respiratory only. I will come to that in one of the slides. When it is from the nasal cavity, then it causes the cribriform plate, goes to the olfactory nerve, and then in your brain, or it can be bacteremia. But no, that is not the right answer. Still better answer, nasopharyngeal swabs or oral swabs, if taken properly, they give you same kind of result. They should be these samples should be corrected, but especially in the set of people when you are suspecting a carrier stage. Okay. So, most of the time I would say in a carrier stage certainly you should collect, but yes, in case of meningitis also in case with CSF and blood culture can also be corrected. So, you are not wrong. What else? Any other sample? One more sample I would like to come from your side. Scrapings from? Yes, not necessarily in pediatric. Anyone in which you get rashes, what we call petechial lesions, they can be collected. So, we are dealing with majorly 4 samples. It could be CSF, the first sample of choice, blood samples, nasopharyngeal swabs, and petechial lesions. So, these are the 4 samples. Now, you have a CSF sample. So, next question is minimum what amount of sample CSF sample is required? 0.5 to 1 ml. Okay. So, that is also, should you try to get more sample? Why not? If you say you have put a lumbar puncture needle and you are drawing very nicely CSF, why you should not be doing? Answer is right, you should not be drawing. If you draw out too much of CSF, what will happen? Have you heard of coning? So, read why you shouldn't draw more than required amount of CSF sample. It should be very clear. Your answer is right, but reasons should also be known to you. So, you have a CSF sample. What you are going to do with that CSF now? You have a CSF. What you are going to do with that? Say loudly. It should be not audible only to me, the last person sitting on the class. Okay, very nice. But before you divide, now I am just interrupting you. The sample has been collected from a place which is 50 kilometers away from the lab. How you are going to bring it? If BHI is not available, if just a vial is available, what you are going to do with it? Not refrigerate? Why? Yes, absolutely right. So, this is one sample which should never be refrigerated, because one of the common cause of meningitis is Haemophilus influenzae, which does not survive at a low temperature. Clear? Very nice. It seems there is only one person who is willing to answer, but no, let others also be. Please sit down. Yes. Any one of you, whosoever is. So, you have 1 ml of sample, for example. What you are going to do with this sample? Say loudly, Baba. You can do. So, whole 1 ml of sample will go into culture, or lesser amount or something? So, question is, you should be asking yourself, what information I am likely to get from this sample, and this stands true for every sample. Just cannot go blindly that I get a sample, I will put into culture. The question you should be asking yourself every time, the reason and logic behind it. So, what all information you can get from the CSF. If you know this, answers will automatically come. I will get answers from you only. If it is meningitis, what is meaning of meningitis? Inflammation of meninges. Okay, fair enough. CSF is in touch with meninges, or yes or no? Yes. If it is inflammation, what type of cellular reaction you are likely to get? What happens in inflammation? Shouldn't be difficult. Say loudly. Neutrophils. Okay. So you are likely to get some amount of inflammatory cells. Let us not go into neutrophils. I will not go at this stage. Otherwise, do we get inflammatory cells in the CSF? No. So you have this answer, and it has come from you only. In case of meningitis, since it is inflammation of meninges, you are likely to get more number of inflammatory cells in the CSF. Okay. How you are going to see inflammatory cells in the CSF? If you put everything in the culture bottle, can you see the inflammatory cells? No. So if you want to see inflammatory cells, how you are going to see? Centrifuge you will do. Okay. No, I can't hear you. By markers. What markers? CD4. No. Why make the life complicated? Where you see inflammatory cells, you routinely, have you seen any time inflammatory cells in your class 12? Sorry? Smear. Under what? Microscope only? So you are going to use microscope. Now, is it always necessary to make an smear to see inflammatory cells? आप लोगों का प्रैक्टिकल हो गया माइक्रोबायोलॉजी में? कोई प्रैक्टिकल अटेंड किया है दोस्त में? यूरिन का प्रैक्टिकल हो गया? What you see, and do you make smears of urine sample? What you do? Sorry? Directly under the microscope. So, can inflammatory cells in the CSF can be seen without making a smear? Yes. So, one thing are we agreeing to this or not, that everything will not go into culture? We are agreeing. So, at least one part you are going to take it out. Okay. What is the normal composition of CSF? So, nothing other than water. See, these are basic. Physiology is why it is taught, why physiology is taught in the MBBS before beginning of this thing, that you should know what is normal, and then you move to physiology to pathology, what is abnormal. So you should know what is the composition of CSF. So CSF will also provide a lot of information about its glucose content, its protein content, and that is very important to know, because in bacterial meningitis, viral meningitis, there is a change in one of the condition, the glucose is raised, in some other condition protein is raised, some other condition nothing is raised. So you should know what is normal composition of CSF, and also to perform analysis on CSF on the amount of glucose, whether it is raised or not, and protein analysis also. So one part will also be required to send to the biochemistry department to know this part. Clear? So now we are agreeing we also need to perform microscopy, we also need to perform biochemical analysis of the CSF, and we need also to perform cultures. Agreeing to that? Okay. Somebody said we will centrifuge the CSF. You only said. So when you centrifuge the CSF, what will happen? Say loudly. Why? So, there will be something in the sediment, and you are very right in saying cells will sediment. And what will be on the top? Supernatant. What supernatant is likely to contain? No, let us forget for a while glucose. That part we have already sending to biochemistry. Pathogens. Okay, not exactly pathogens, but what we use it is the antigen detection test. Okay, so the CSF sample is divided into 3 parts. One part is for performing you centrifuge it, you have sent also to biochemistry. The sediment is used for performing smears, and you see whether any pathogens you can see, gram stain you can perform, and also directly if you perform a drop of CSF, you perform microscopy whether the number of pus cells are increased or not, because they are indicative of inflammation. Supernatant is used for performing antigen test. One part which has not been refrigerated and should not be refrigerated, preferably if it is from the far away place, it should be in the BHI broth, as very rightly said by your friend, and put into the culture. What now, but I said 3 parts. So, what happens to the third part? You have 1 ml, you have already divided 0.3, 0.3, another 0.3. So, another 0.3 is with you. It is said that this 0.3 microliter of sample should be added to BHI broth and incubated as a backup broth. Okay. And it is found that many times when the culture yields are negative from the directly from the part which you have already inoculated, this backup broth can give you sometimes a better culture yield. So, your CSF is going to be divided into 3 parts. Understood? Sit down. Next thing is, so next comes to the blood sample. Though it is very generic question, but when you would like to collect the blood sample, preferably before giving the antibiotics. But very important thing to understand, in other diseases we say hold your antibiotics till the culture reports come. But in life threatening conditions like meningitis, encephalitis, wherever life threatening, you should start with a broad spectrum antibiotic, wait for the culture reports, and then you can taper down or scale up your antibiotics. So, you will not wait for culture reports in such life threatening cases, rather you would start on those antibiotics based, if the institute is good and they have their antibiogram and you have a local data available, your antibiotic choice should be dependent on that. So, keep in mind what are the pathogens responsible. CSF will obviously the choice to collect, other samples will be blood, petechial lesions also we said, and nasopharyngeal swabs, as the girl there sitting there told us. This is what we have already discussed in detail. Minimum 1 ml of sample or 1 ml is the optimal choice. Amount of sample must not be refrigerated, do centrifugation, supernatant and sediment we have already discussed what should be done with those samples, and then your gram staining, superformed sediment. And now question comes, what are the medias to be used? Why it is important? The first thing what we said in this slide is, keep in mind the organ is causing meningitis, because as you had said, and mistakes are, it is good to make mistakes in the class, unless otherwise you will not learn, and you will not make mistakes in the exam if you do not understand. Now, suppose I go with your choice E. coli, also it causes meningitis, no doubt about it. What type of media I will require for growing E. coli? Any media will do good. McConkey will do good, nutrient agar it will grow, everything, because it is not a fastidious organism. But if I am thinking and I have this knowledge that the pathogens which can cause meningitis are the most common cause of reasons of causing meningitis are pneumococci, Neisseria meningitidis and Haemophilus influenzae, and all of them are having a little different growth requirements. Then my choice of media will be dependent on the pathogens which I know. That is why it is important to keep in mind the organism which is causing meningitis. So, now what media will be used? If suppose now you have 3 bacteria we are talking about, primary pathogens, the class is all about. So, primary pathogen is, first is pneumococci. What media I will use? Blood agar. Fair enough. Next pathogen was Neisseria meningitidis. What I will use? आपके हाथ में क्या है भाई? मोबाइल लेके हाथ ना बाहर निकल जाइए. अगर क्लास में आपको मेरे पढ़ाई के बजाय मोबाइल देखने हैं, गेट आउट. Yes. So, if it is Neisseria meningitidis, what should be the choice? Sorry? Thayer-Martin. Okay. Haemophilus influenzae? Chocolate agar. Why? Okay. So, you are saying factor NAD, factor 5 and 10 is required. Good enough for you. Now, if suppose though you have covered chocolate agar here, but there is a possibility the pathogen can be something else other than these 3, because I always say that patient doesn't come with a placard saying that I am suffering from this. So, these are the most common pathogens, but it could be anything else also. How you are going to ensure that anything else other than these 3 will also grow? Is because you have chosen a chocolate agar and blood agar which are already there, and they will support growth of practically all other non-fastidious organisms which may be reason for causing meningitis. Okay. So, do not go always with the selective medias. There has to be 1 or 2 media which will support in general growth of other bacteria also. So, because Thayer-Martin is very specific for Neisseria. So, if you are using only Thayer-Martin, you will miss out every other pathogen if Neisseria is not the cause. So, we have taken these choices, which is chocolate agar, blood agar, modified Thayer-Martin, and as we said that the third part should be as a backup as a BHI or in a glucose broth. Now comes question, what you are going to expect to see now? You have the culture, and you must perform from the supernatant the antigen detection test, which will give results far earlier than the growth of culture. What you expect to see now from these? One by one we will go. If suppose you are having a pneumococci as the pathogen, what exactly you would see in the plates? If pneumococci is the pathogen, what you will see? Yes. Alpha hemolytic colonies. So, we will skip these slides because we have said blood, Patechi, these samples we have already taken into these are the Patechi, how they look like. Biopsies can also be taken, nasopharyngeal swab can be taken, autopsies can be taken. Okay, one thing which I forgot, where you will incubate? 37 degree, no doubt about it. Preferably 5 to 10% of the CO2 is preferred. It is not going to harm any other pathogen, but it will support growth of Haemophilus, even the Neisseria. So, incubating conditions are these. Now comes what you are going to see. So, if it is pneumococci, what you are seeing in these 2 culture plates? That this is the blood agar. There is some greenish discoloration is here. There is one disc also, this we will discuss, but then you are going to see alpha hemolysis. Okay. So, alpha hemolysis means you are dealing with a pathogen which causes alpha hemolysis. Will you perform a gram stain from the plate? Yes. In gram stain, what you are likely to see? Gram positive cocci arranged in pairs. If you do not see pairs, but you see gram positive cocci and alpha hemolytic. Now, because at times you may not see that classical arrangement what the book says, that they are lanceolate shape, arranged in the cocci, capsule is around, but you will see a gram positive cocci which is alpha hemolytic. Will you jump to the conclusion that it is alpha hemolytic, it is a pneumococci? Sorry? It could be Streptococcus viridans also. And will you treat that? Will you treat that? Probably no, because viridans group of Streptococci is known to be commensal flora. But you should not completely discard it, because this is a sample, CSF sample or blood sample, which are otherwise sterile sample, and we presume that the samples have been collected very aseptically without any chance of contamination. So, you should not just discard the sample if you are seeing gram positive alpha hemolytic, but then you should be little careful and take the findings with a pinch of salt. But if it is alpha hemolytic and you are classically seeing a picture of gram positive cocci in pairs, and then you are thinking it is presumably pneumococci. So, how you are going to confirm if it is pneumococci? इनका प्रैक्टिकल न्यूमोकोकाई का हुआ है, एक काज है. Okay. So, you have a practical class also. So, your job now here is to differentiate alpha hemolytic gram positive cocci, whether it is pneumococci, which is established pathogen, or it is a viridans group of streptococci, which is a commensal flora. So, how you are going to confirm are by 2 or 3 things. One is this picture you also saw in the media plate, blood agar plate, there is a disc around it and there is no growth. It means this particular disc is inhibiting. What this disc could be, especially those who have attended the practical class? Optochin. So, pneumococci is sensitive to optochin. Here it is, there is no inhibition of growth in the other side of the plate. So, optochin sensitivity, that is one thing, and I will show some slides. Bile solubility and inulin fermentation, that is also we will deal with it, and these are confirmatory tests. Another quality of pneumococci is that they undergo lysis by the bile salts. Means if you add bile salts are 10% of solution of deoxycholate solution. I think I have a picture here, I can, yes, this is the one. What you are seeing in these 2 test tubes, there is a one test tube where you are seeing a homogeneous suspension and another tube in which there is a clearing on the top and then some sedimentation has taken place. So, deoxycholate solutions, what do? They act as a surface active agent and make the streptococcus cells to undergo lysis. Once they lyse, the cells will or the degraded cells will sediment in the bottom. So, I can skip now some of these slides. Yes. So, you are going to confirm they are gram positive, alpha hemolytic, sensitive to optochin and bile solubility. This will confirm your identification whether it is pneumococci or not. How do they look like? You already have answered, they will be gram positive cocci in the pairs. What other infections does it cause Streptococcus pneumoniae? The name itself says what? Pneumonia. What else? Children will often come with the sinus or they will come with the earache. So, middle ear infections, pneumonia, paranasal sinusitis, these are all common features. Always remember, meningitis is usually a secondary event. not the primary events. So, secondary to any of these things, it could be pneumonia or middle ear infection and you get meningitis. Fine. So, I will skip this. We have already known that this is a fastidious. Okay. Now, suppose in the CSF sample, we performed a gram centrifugation and you see the slide, and if you see this picture, now please describe what you are seeing in this picture. Answers will come from you only. If you just see the picture and tell whatever you see, don't go into complicated things. Say loudly. You are seeing leukocytes. Are they multinucleated or multinucleated? So, if they are multinucleated, what type of cells they are likely to be? Neutrophils. What else you are seeing? They are gram negative cocci. Some of them are extracellular and some of them are intracellular. Okay, now we usually say meningococcus or gonococcus are intracellular, but this is true but not always seen in the smear. So, if you end up seeing extracellular gram negative cocci also, do not say no, since it is extracellular, they are not going to be meningococci. So, if your mind knows, eyes will see. So, in this picture you can very well make out that there are polymorphonuclear cells and you are seeing gram negative cocci. If this is the picture which you are seeing in a CSF sample, which is supposedly a sterile sample, it is a very big lead to understand, yes, you are dealing with a case of meningococci because it is a gram negative cocci. You have plenty of gram positive cocci in life, but gram negative cocci are very rare, limited numbers. So, that is one very strong lead to think that this is a meningococcal meningitis. Fine. Okay. What you see, please sit down. Now, you have a chocolate agar plate which exactly looks like Thayer-Martin plate. So, not such any difference you will see, but you are going to see these type of colonies. Okay. I said seeing a under microscope is a big lead, but if you wish to confirm whether it is a meningococci or not, at least what you should be doing it. Perform a gram staining from this, that is one thing. What you are likely to see? Gram negative diplococci. Okay. So, we will skip to, we will see something like this. This is a cell which is not a nucleus, it is epithelium, but then you are seeing a gram positive diplococci. I never go say that you should always look like kidney bean shaped and they are opposed on axis to the long, because these are not always seen. They are seen, but not always seen, and if they are not always seen, the typical picture is not always seen, you should not say no, I am not dealing with this. But you will certainly see gram negative cocci which is there. Okay. What other test we will perform is your oxidase test. This is one gram cocci which is oxidase positive. All other gram positive cocci are oxidase negative. This is gram negative cocci and also a oxidase positive. That should be a good amount of confirmation that you are dealing with a case of. In how you perform oxidase test in the practical class, they will be telling you. We said antigen detection test, and from the supernatant, don't write everything is given you, listen to me. So, the supernatant is going to be having antigens, and if you have antisera, you should go for either A, B, C, Y types. Negative test does not mean that you are not dealing with a meningococcal meningitis. Okay, remember this. Positive test, yes, it is a very again lead to your confirmation. Negative does not mean that you are not dealing with a meningococcal test. So, you perform antigen detection test, you may perform serological test like ELISA or something, but their availability of monoclonal antibodies and ELISA test is a limiting factor. I will just want to tell you, yes, this is the picture which I was looking for. So, typically how the Neisseria is spread. So, it is a respiratory, upper respiratory, it crosses your mucosal epithelial cell barrier and then blood vessels and then to cerebrospinal tissue, that is the. Now, meningococci meningitis is all, there are other diseases also which we should certainly make a reference to it, is what is meningococcemia. What does it actually, this word would mean? Meningococcemia means, mia means it spread in the blood, meningococcemia means the blood borne. So, meningococcemia is a very dangerous entity and it is also seen. So, what are the predisposing factors? Crowded conditions, people who are anywhere crowded, it could be hostels, it could be crowded jails, military barracks and so on. And there is another thing which we should know, meningococcal infections, especially the repeated meningococcal infections, are associated with deficiency of complement system C5, C6, C7. So, if a child or a person is repeatedly getting, you should certainly look for underlying facts whether I am having these deficiency of terminal complement system or not. So, that is another very important part for you to understand. SLE, hepatic failure, myeloma, any of these predisposing feature can be there. I will forget this. So, there is meningococcemia and there is a fulminant meningococcemia which is known as a Waterhouse-Friderichsen syndrome. So, if you get a short note by either of this fulminant meningococcemia or Waterhouse-Friderichsen, the examiner is intending to ask you about meningococci. Next part is, if suppose you are, this we have all discussed, oxidase positive. Now, if you see in your culture plate or in the CSF this picture, what you are seeing here? What you are seeing here in this picture? Say loudly, but it should not be a rocket science. I am asking you to describe whatever you are seeing here. Yes, madam. Say loudly. Different sizes of the bacilli. Are they gram positive or gram negative here? Gram negative. So, you are in this picture you are seeing gram negative bacilli which are of different sizes. What is the term used for this? Pleomorphic. Already it is written also here. So, if you are seeing a pleomorphic gram negative bacteria in the CSF smear, what you should be suspicion now? And it is common sense. We talked about 3. We already said in meningococci what you will look like, gram negative cocci, gram positive cocci will be pneumococci. So, third one remaining is Haemophilus. So, it is a case of mistaken identity. Why? Because previously the disease was thought, the Haemophilus influenzae, that it is a viral disease and influenza virus, but then it was realized no, this is a bacterial entity and reservoirs are only humans. This shows lot of seasonal variation, typically in the middle of mild winters kind of months. And what you should be knowing here is, I will ask you another question here now. Which vaccine do we give against Haemophilus? Which vaccine do we give? HIB stands for Haemophilus B, and it is, so previously we used to give DPT, trivalent, now we give pentavalent vaccine. So, what has been added to it? No? No? So, you will find out what is pentavalent vaccine given. One is added is Haemophilus influenzae B. Why type B? Why type B? Answer lies here, that out of A, B, C, D, E, whatever types are, it is the type B which is the most common disease. And this hepatitis, the why we give is because infection with hepatitis type B, 50% infections are causes meningitis. Rest, you are not much worried about pneumonia, epiglottitis, cellulitis, but because this is a very life-threatening illness, that's why Haemophilus influenzae type B Hib vaccine has been added to it. So, as we said, this is gram-negative, aerobic, cocobacilli, pleomorphic, and another bacteria which is oxidase positive. It won't grow on McConkey agar. It won't grow probably on the blood agar also. And you very rightly said that initially you made a choice of chocolate agar because it has a factor X and V. What are those X and V? Right. So, it is characterized by its requirement for factor X and V, and this bacteria do not survive refrigeration. So, you were right in saying that CSF sample should not be refrigerated at any time. So, these are 6 different serotypes A to F, and 95% of invasive diseases caused by Haemophilus influenzae type B. That is why vaccine is covering Hib, and it is likely to take care of, or it takes care of 95% of the cases which are caused by Haemophilus influenzae. So, how you confirm whether this is Haemophilus influenzae? How you demonstrate that this is requirement of factor X and V? So, one thing is, you have any idea? Satellitism, that is one answer. Any other way of doing it? We will come to satellitism in a while. Any other particular way? If you keep a source of factor X and V on a blood agar plate, the bacteria will grow near to the wherever the source is. So, that is one way of doing it. Second way is very likely satellitism. So, would you care to explain what is satellitism? This is picture. Now, if you can tell me with this picture how you perform or how you demonstrate satellitism. Staphylococcus aureus. Okay. How it will produce? What Staphylococcus will do actually on a blood agar plate, that you should know. It will lyse the RBCs, complete hemolysis, so the factors would be released. Okay. Not it is that it produces. Okay, then not around it, on a perpendicular to the line of inoculation of Staphylococcus aureus. Okay, then why it will grow? Yes. So, because factor X and V, the RBCs are lysed, that will be the area where the factor X will be much more than as you go distantly. So, the colonies will be more and bigger nearer to the Staphylococcus streak, and as you start going farther away, you will see probably no growth or very small colony as you see the satellites. That is how why it is called satellitis. Good. So, that brings me to the end of this class. So, what you have learned is primary pathogens, primary bacterial pathogens are pneumococci, Neisseria meningitidis, and Haemophilus influenzae. What would be your choice of samples and how you culture it? And not to forget, I hope you will not forget that it is not whole ml, 1 ml will go into culture, rather it will be divided into 3 parts. A very important hint or lead will also come from the relative concentrations of protein and glucose in the CSF, that will tell you whether it is viral or bacterial, and then confirmation by these. Okay. Any questions? Feel free to ask. आप attendance ले लीजिए.